Thank you. Yes, you're going to hear a lot about axons. So in this first slide here, actually, I show some axons here coming out of the microgrooves of a device that I will describe later. Microfluidic culture of neurons, and the axons can be separated from the cell bodies. Now, in the animal or in humans, the axons are not naked like this, but they are surrounded by myelin. And myelin in the central nervous system is a product of a cell, a very specialized cell called the oligodendrocyte, which extends several cytoplasmic extensions that wrap themselves around axons to form the myelin sheath. Those myelin sheaths are essentially devoid of cytoplasm because the two extensions of the plasma membrane come together and all the cytoplasm is extruded, except in some cytoplasmic channels, which are highlighted here, where there is continuity, complete continuity between the cytoplasm of the oligodendrocyte and the myelin sheath. And some of those channels are, as you can imagine, in direct contact with the axon. Now, the function of myelin is actually twofold. One that we all know is electric insulation, and this is the reason for saltatory progression of the nerve impulse. One which is less appreciated, or was, and now it's more and more appreciated, is that the myelin is essential for the maintenance of the axon. If the myelin suffers from mutations, for example, which disrupt some of the myelin protein or other causes, very often the myelin may persist as a structure, but the axon will degenerate. And this is the case, for example, in several human diseases, such as some of the Charcot-Marie-Tooth genetic disease. I won't elaborate on all of that because of the interest of time, and I will come straight to Theiler's virus, which is a cardiovirus, so it's a picorna virus, very closely related to a poliovirus in humans, which is a mouse virus. It infects mice in nature. It's very, very common in wild mice. And in the lab, this virus is used for, it's also like many picorna viruses transmitted by the oral fecal route. But in the lab, we are interested in the neurological disease that this virus causes, and therefore we inject the virus directly into the brain of the mice. And there is a biphasic disease that ensures this inoculation. The acute disease lasts about 2 weeks. It's an infection of neurons. And then, if the mice are genetically susceptible, and I won't elaborate at all about this aspect in this talk, the virus may persist for the lifetime of the animal, but it does not persist in the neuron. It persists in oligodendrocyte, the one I just described to you, some other glial cell, including astrocyte, and also in macrophages, which invade the CNS due to this persistent infection and are part of chronic inflammatory response for this disease. This disease, by the way, has been used for many years as a model for MS, for multiple sclerosis. Now, the question I'd like to address here is how does the virus traffic within the CNS? I say it infects neurons first, and it persists here for a lifetime in some mice which are genetically susceptible. How does it go from the neuron to the astrocyte, to the oligodendrocyte, astrocyte macrophages? Well, we know from a lot of work, some old, some more recent, that Theiler's virus is transported into the axons. This is tissue culture neurons, and you can actually, and this is detecting the capsid of the virus, and you can follow the capsid all the way down some axon, which can be also characterized by double labeling with axon-specific markers. So we know the virus traffics down the axon. How does it go from the neuron to the glial cell? Well, the question that we asked and tested is, could the virus go from the axon into the myelin, which surrounds the axon, then spread through those cytoplasmic channels to the cell body of the oligodendrocyte, and then from there to macrophages, probably after oligodendrocyte cell lysis or even without lysis. And we decided to test this hypothesis using a mutant mouse, which is called the shiver mouse, because this mouse has oligodendrocyte. Actually, it has a probably increased number of oligodendrocyte in its white matter, but there is no myelin because of a deletion of one of the most abundant myelin protein. The basic myelin protein is deleted. So the mice have oligodendrocytes, they don't have myelin. The prediction is if indeed the virus was spreading from the axon into the cell body of the oligodendrocyte through the myelin, there may not be any spread in those mice because the myelin is missing. This is a cartoon of this hypothesis, a normal mouse. The shiver mouse, there is no myelin, and would the virus be prevented by this lack of myelin to spread to the cell body of the oligo and to the other glial cells? So to test this hypothesis, we use an assay which consists in injecting the virus in the eye of the mouse, where it's going to infect the neurons which are in the retina, the retinal ganglion cells, and those neurons send their axon down the optic nerves. In the optic nerve, there are no neurons at all. It's pure axon, myelinated axons. And this is a sort of two-chamber compartment. There is no physical connection where the virus could diffuse between the vitreous of the eye and the optic nerve. So if you infect the optic nerve, you expect the virus to travel down the axon that I showed you before, and if it's going to cross from the axon into the myelin and the oligodendrocyte, you should be able to observe that in the optic nerve. And actually, by not infecting the contralateral eye, you have here negative control because the virus could spread via the bloodstream, for example, to the whole body of the mouse, including through to this contralateral optic nerve. But if this contralateral optic nerve is not infected, it will be a proof that the infection in this side is coming from the ganglion, the retinal ganglion cell from the eye which has been injected. So this assay, therefore, consists in injecting the virus here, looking on the optic nerve, and to make sure that the virus has traveled from down the optic nerve, you can look at the ganglia in the brain where the optic nerve connects, which is the LGN and the superior colliculus, and actually you can see that the virus has arrived at the end because some of the neurons are infected. For some reason, one side just disappeared here in this computer, but this showed that the infection of the retina. Actually, here it shows it in more detail. Those are retinal ganglion cells, which can be characterized by a marker, which is called V-GLUT. This is the virus which infects those same cells. And the virus arrives at the end here, and you can see that along the way in the optic nerve, there are some infected cells. This is, again, staining for capsid antigen, and some of those cells are oligodendrocytes. Now, how do you characterize a cell? How do you quantify everything? Well, it can be done by doing a whole mount immunofluorescence on the optic nerve. The optic nerve of a mouse is not a very big thing. So antibody, using detergent and a few tricks, can diffuse quite easily through an optic nerve and will stain, and this is staining with anti-capsid antibody, will stain cells which have been infected. And you can see here a typical oligodendrocyte which has been infected with a cell body, and you can see the cytoplasmic extension down the myelin surrounding axon, which contain also a viral antigen. And this is a very different cell type. It's an astrocyte, which is also infected. So an optic nerve of the mouse can be scanned down the microscope very quickly for the whole mount, and you can count the number of cells which have been infected or not infected and determine their cell type just by morphology. We've actually made sure that this is relevant or reliable by doing some double staining, but it can be really done just by morphology. And what happens is, after checking that replication in the retina is the same for the mutant and for the wild type, that the virus travels at the same speed because it arrives on the other end in the same way, you can see that there is a big difference between the infection of oligodendrocyte in a wild type mouse and in a shiverer. So therefore, the hypothesis which I proposed is probably correct, that the virus used the myelin to travel from the axon into the cell body of the oligodendrocyte, and from there probably into the astrocyte. Now, what is the mechanism by which the virus traffics from the axon into the myelin? This virus is a picornavirus, and picornaviruses have the reputation of being very cytolytic. So one first question is, probably those neurons are going to die at some point, and is the axon lysed by the death of the neuron, and is axon lysis required for the virus to be released and to infect the myelin through some kind of receptor-dependent entry? And to test this hypothesis, we used a mouse, a mutant mouse, which is a WLDS mouse. This stands for Wallerian slow degeneration, Wallerian degeneration slow mouse. This is a remarkable mouse, which is well studied by neurophysiologists, because the axons resist all sort of toxic aggression to the cell body of the neuron. Actually, the axon can be separated, can be cut in culture from the cell body, and those axons will continue to function and to carry organelles and to conduct influx for hours or days separated from the cell body of the neuron. And the reason for that has been investigated. The mutation has been well described, and I won't have time to go into the detail of all of this. So these mouse mutants allow us to infect the retina of the eye of those mice in wild type or in the WLDS, and then to look into the optic nerve. Here in the optic nerve, you can see that some of these axons degenerate in the wild type, they swell, etc. But there is no evidence for degeneration in the WLDS because the axons are so resistant. And yet, the virus is able to traffic almost as well in terms of number of oligoendocytes infected, for example, in the wild type and in this mutant. So lysis of the axon is not required for the virus to go from the axon into the surrounding myelin, which is a remarkable property for picornal virus. So what is the mechanism? And at this point, I don't have the answer. We're working on those various hypotheses, which I won't detail all of them, but I want just to point out to this scheme here, which is derived from the information existing in the literature that myelin, not only, as I said, is an insulator for the axon, but is also supporting the axon. And one of the functions of the myelin, which has been described in invertebrates, not totally characterized for vertebrates yet, is that the myelin plays a role in cleaning up unwanted organelles or unwanted material from the axon. And this is probably because you can imagine some axons in the body of mammals, for example, including us, which are 1 meter long. Now, if all the unwanted organelles, including mitochondria, which needs to be degraded, have to be transported retrogradely all the way to the cell body of the axon to be degraded, the volume of the cytoplasm in the axon far exceeds that of the cytoplasm in the cell body. And there is a number of problems there. So it's been hypothesized that one of the functions of myelin is to clear those unwanted material along the way from the axon and to therefore lighten the work of the cell body of the axon in recycling all of this. So one of the hypotheses is that maybe the virus has found a way to introduce itself in the reverse direction in this pathway and to use this communication that exists between the cytoplasm of the axon and the cytoplasm of the myelin to traffic without lysis of the axon. This is just to distinguish, and I won't elaborate because of the time, that it could be virus with their capsid, which we detect in the axon, as you've seen in culture at least, that is transferred and which would require probably a receptor to go in. But it could also be replication complexes that would leave the cytoplasm of the axon to enter the cytoplasm of the myelin, the cytoplasmic channel in the myelin, and this would be capsid independent. And we don't know yet at this point, but we are working on those issues. Now, to go further in those issues, it becomes a bit difficult to use the mouse. And this is where I'm going to introduce the microfluidic chambers and the culture of neurons in those chambers. For those who don't know those devices, they are microchips in a way, consisting of wells, which you see here, for example, 4 wells, into which you can introduce cells, viruses, media, whatever you want. And those things are connected on this side, the 2 gray chamber, and on this side, the 2 white, by a channel which is under the surface of the chip, and then, which is here, and here, between the 2, there are some microgrooves, which are very small, 3 microns tall, which connect the 2. And the length of those microgrooves can be adjusted according to the need of the experiment. And when you grow axon in one of these chambers, axon loves to find, you grow neurons, in one of the chambers, their axon love to find those channels, and the axon will go in the channels and come out on the other side. Now, if you keep a slight difference of volume between the two chambers, you can completely isolate the two media, and nothing, for example, which is in this chamber will diffuse back into this one, so that you can separate cell body from axon terminals in terms of using viruses, using inhibitors, using whatever you want to use. And what we aim to do is to culture neurons, for example, in this chamber, have them extend their axons here, and then you can add oligodendrocytes in this chamber that are going to extend myelin to contact these axons. And because of the isolation, you can introduce a virus here. You can use two sort of tricks to try to understand what's going on in terms of going from the axon into the myelin. First of all, you can use neurons, primary neurons, of course, they are primary cultures, and oligodendrocytes coming from different genetic background, having mutation in one gene or mutation in another gene. You can use inhibitors that you can put to the cell body of the axon. You can use inhibitors that you give to the oligodendrocyte to try to decipher this mechanism. Now, this is ongoing, and I don't have a result to give you on this aspect. We can myelinate, or at least have myelin contact axon in those cultures, but I don't have a result to tell you about. But this gives me my transition to the problem of Parkinson. Oh, sorry, this is another illustration of this, which shows the microgroove here, and you've seen that in my first slide. Here you see the soma of the axon down the microgroove, and the axon coming on the other side as stained by calcine AM. So in conclusion, Theiler's virus infects neurons for 2 weeks, persists in oligodendrocyte macrophage. The virus infects myelin oligodendrocyte, comes from the axon. Axon to myelin traffic doesn't require axonal degeneration. And we hypothesize that the virus takes advantage of pre-existing communication between the two traffic. And I didn't elaborate at all on that. But if the virus is not able to move to oligodendrocyte, it is cleared by the immune response. So this step is essential to allow the virus to persist. Yes, so the microgrooves give me my transition to the story of Parkinson's disease, because when we were, and when I say we, it's Eric Freund, a postdoc, who's been doing all this work on microfluidics. When we were testing the system and setting it up, it came to us, and this is very serendipitous, and then I won't go into the exact reason why I got involved in Parkinson's disease. It's really totally a coincidence, which has to do with Air France losing one of my luggage. But I can detail that in the question if you're interested. So Parkinson's disease, as one aspect of Parkinson's disease, we thought could be addressed using those microfluidic chambers. Parkinson's disease, as you know, is a very common disease, unfortunately. It's characterized by the loss of dopaminergic neurons. And what's really interesting or important is that Parkinson's disease follows with time a progression and a diffusion within the CNS, which follow axonal projection, both retrograde and anterograde. So the lesion starts here, and then with time they move to the striatum, and then they move to the nucleus accumbens, and then they also move to cortex, including frontal and prefrontal cortex. And this is actually the reason for the fact that some patients develop dementia, and Parkinson-associated dementia is a very serious problem. So there is this progression, which has been well described by several others, including Brahic in Germany, staging the disease. And actually, the first lesions, which are not illustrated here, are found in the olfactic bulb and in the gut. And it appears that then they spread from there into the rest of the CNS. Now, the characteristic of Parkinson's disease is those inclusions, which are in neurons, which are called Lewy body. Now, Lewy body is made of an aggregate of insoluble protein, several of them, but the major protein in those Lewy body is alpha-synuclein. And alpha-synuclein is both a synaptic and a nuclear protein, hence the name synuclein. It is a protein which is alpha helix rich when it is bound to membrane, but it can acquire a beta sheet conformation and then aggregate into fibrils. And there has been quite a bit of discussion in the literature, including from Stan Prusner, that Parkinson's disease could be a prion disease. And Parkinson's disease, therefore, could be, the hypothesis here is Parkinson's disease could be an infectious disease in the sense that it could be infectious within the organ, but we have absolutely no evidence that in Parkinson's disease, infectious between individuals. It's just infectious in the way it would spread within the body of the patient. So the question is quite relevant for the following reason, which is some patients, and actually quite a large cohort of patients now, have been treated, or there is attempts to treat them by grafting some neurons into their brain, and it's been observed in some that died years later that the graft has been invaded by those Lewy body. So it's quite clear that this Lewy body spread, and they can enter a new cell. So how do Lewy bodies spread within the CNS? I told you that they appear to spread from the pathology along a neuronal pathway. And we decided to test this hypothesis with the microfluidic chambers, which I just described. So, one slide disappeared here. This was just to show you that to do those experiments, of course, we need insoluble alpha-synuclein. So through a collaboration with a colleague of mine in Paris, Ronald Melky, who is a protein person and a yeast prion person, we obtain insoluble fibrils of alpha-synuclein, which can be heavily labeled with a fluorescent dye, such as an Alexa dye. So we have some highly fluorescent alpha-synuclein fibers. And when we give them to neurons, in fact, indeed, some of those fibers enter the cell body of the neurons, as you can see here. The neurons are characterized with beta-tubulin, which is tubulin-specific, the beta-3 for neurons. And so indeed, those fibers of alpha-synuclein are able to enter the neurons. And the neurons support that for quite some time. Eventually, they die. But since they die, in any case, in culture after some days or weeks, it's not all that clear. It's probably toxic to the neuron after some while. But they survive after they gobble up the alpha-synuclein fibrils for quite some time. You can see it here at higher magnification inside the neurons. As you can see, there are some cells here which are not neurons. They are astrocytes, actually, because those primary cultures are always cultures of both neuron and astrocyte that come together. And they also internalized alpha-synuclein, but will be interested only in what's happening to the neuron. So first thing is, in the microfluidic, and this is not very clear from where I am, but I don't know if you can see it from further back. Well, the first question is, do we see the alpha-synuclein fibrils within the axon in the microgrooves? And sure, we did. It's not very strong, but we have some punctae of fluorescence which correspond to the Alexa 488, which is a synuclein, in the axon. And to make sure it's not background, we look at the spectra of emission of those Alexa green dots, and they coincide the spectrum. This is the spectra for the CTB, which is a cholera toxin B label with Alexa 555, which stain the neuron and the axon. And this is the spectra for the alpha-synuclein, and the spectra for the alpha-synuclein alone, and the green dots in the axon coincide. And this is what would be autofluorescence. So we are convinced that indeed there is alpha-synuclein in the axon. Now, is alpha-synuclein really transported? And also, is alpha-synuclein eventually delivered to another axon, another neuron? And the question is it transported retrogradely, anterogradely? So with this device, having the neurons here, sending their axon here, you can put neuron also on the other side and study all those questions. And here it shows that if you put the fibrils here, they're going to show up in the axon here if they are transported retrogradely. And if you look at this situation, you may be able to show transfer to the second neuron. And indeed, this is what we found. We observed that indeed the alpha-synuclein is very efficiently transported retrogradely. So to show if it was anterogradely transported, we plate a neuron on one side, wait until they send their axon on the other side, then plate a neuron on the other side, didn't give them time to send their axon, and added the fibrils of alpha-synuclein in this side, and ask, are we going to see alpha-synuclein in the cell body of the second neuron? And indeed we did. This is a control for the retrograde. There was no retrograde transport, and we could detect alpha-synuclein in the cell body of the second order neuron in those chambers. Now, this is, of course, the same alpha-synuclein which had been introduced in the cell body of the neuron, which is miles away on this scale, and which has been transported on the axon. There is no amplification in this system. So although it's only the inoculum, so to speak, we can detect some here. And if it is a real prion disease with amplification, it's easy to understand how the pathology could spread. So in conclusion, and I am done, alpha-synuclein is internalized by neuron. It's transported both anterogradely and retrogradely. It's delivered to the second order neuron, and Parkinson's disease could be a prion disease with spread of the infection via axonal projection. I'd like to thank many collaborators. Eric Freund did all the work on the microfluidic, which I just described to you. Some of the work on Theiler's was done in my previous lab at Pasteur in Paris. Ronald is the one who makes the fibrils and labels them with Alexa. And above all, I'd like to thank Carla Kierkegaard for her wonderful hospitality at Stanford. Thank you very much.
We'll have time for one or two questions.
The axons in the shiver mouse, are they full of more cellular junk because they don't have the myelin around them?
Yeah, very good question. They eventually degenerate. I don't know if I can say that they are full of cellular junk. I don't know if that's been clearly shown. But there are several mutations, and the shiver is one, where the axon, the shiver has a very strong phenotype. I mean, it's a very sick mouse. But the axon look fine until the mouse is almost a year old or 9 months old. And then there is not only myelin loss, I mean, there is no myelin, but there is also axonal degeneration. Interestingly, in some different mutation, like some PLP mutation, where the myelin is still there, but abnormal, axonal degeneration takes place even sooner than in the shiver. So the axons survive and function for some time, and then eventually they die. Whether they accumulate junk, I cannot tell you.
Kind of consistent with the hypothesis still, though.
I'm sorry?
It's consistent with that hypothesis.
Yeah.